Alright, so this is just a quick overview of what you need to be able to do with prokaryotic cells. What I've listed here are 4 separate command terms for IB: draw, label, annotate, and identify. And these are all really great to be able to think about and use for this drawing of a prokaryotic cell, because you have to be able to do each one of these 4 things. And if IB asks you to be able to do one of these, you need to know what they're asking for and be able to give it to them so that they can give you points. So it's a little give and take. It's a feedback loop. They tell you what they want. You need to give it to them so that they can give you the points that they're asking you for. So what I'm going to try to do is sketch out a diagram as best as I can. So we've got a couple of components to our prokaryotic cell, and all I'm doing right now is drawing it, and not very well, but it doesn't need to be a great drawing. It just needs to have the components that we need to be able to know and talk about and annotate. So what I've drawn here is the nucleoid region. Let's say that we've got, let's pick a good color here. Alright, and I'm going to draw some ribosomes in here. Now, I'm going to talk a little bit about, I'm going to label my drawing here, but I'm going to talk a little through the annotation that you're more than welcome to write down, but you'll get the idea of what should be added if they ask you to annotate. So I'm going to label in white here. So we've got a cell wall on this prokaryotic cell, and that kind of provides some structure, but know that this is made with peptidoglycan, not cellulose or chitin. Cellulose is the cell wall of plants, and chitin the cell wall of fungi. In the prokaryotic world, we're talking about cell walls that some of them have peptidoglycan, some of them don't. We're talking about an E. coli cell. We've got peptidoglycan. We've got our cell wall. We've got our plasma membrane on the inside, also known as the cell membrane. We've got some pili on the outside here, and these pili are going to be used to attach one bacterial cell to another in preparation for exchange of DNA, which kind of counts for sexual reproduction because it is exchanging DNA. We've got our nucleoid region, or free-floating DNA. There's no proteins that they're bound around. We've got here's a ribosome, and remember, ribosomes are made out of ribosomal RNA or rRNA, and they make proteins. We've got our circular DNA here, which is not necessary in the normal operation of the cell, but can help the cell adapt to new environments. And we've got several flagella here which help in the motility. So what I've done is drawn our prokaryotic cell. I labeled it, and what I talked about with each of these organelles, it would definitely be important for you to add if you were asked to annotate. And the identify would simply be if you were given an image of a prokaryotic cell, be able to identify these components to it. And usually what IB is going to be asking you to identify excuse me, what to identify are these structures on an electron micrograph. So you might want to Google and look at some images of electron micrographs of an E. coli to kind of practice a little bit so that you're able to identify these components well. And that really covers much of the prokaryotic cells and what you need to be able to do with them, but by no means covers all of the things that you need to know regarding prokaryotic cells.